And so these don't have an unequal sharing of electrons. Their sharing is equal, whereas these guys are unequal. This guy has more of the electrons around him than this guy does. And that's why they get the charge. These guys don't have that going on, so they don't interact very well with those. So when we're talking about triglycerides and fatty acids, triglycerides and phospholipids, this whole thing is nonpolar. So if you take some some triglycerides, take some out of my stomach, drop it in water, they're not going to interact very well. However, in this phospholipid, this end's nonpolar, but this end's polar. So this end wants to interact with the water. It's attracted to it. It's hydrophilic. Have you heard that term?
Yeah, the water loving.
Yeah. Whereas this end's hydrophobic. And that becomes really important in the next section when you talk about the biphospholipid layer for the cell wall. Because these guys bunch together so that these fatty tails can be on the inside away from the water, whereas here's the outside of the cell, the inside of the cell that both have water. So the heads can be facing the water where the tails are facing away.
The tails basically give the structure then, so that's why the cell doesn't break down or anything.
Yeah. Yeah. And it also makes it so that some things can pass through and some things need help passing through, like glucose needed that transporter that I talked about earlier to pass through. Whereas those steroids don't need help passing through because they are hydrophobic. So they interact really nicely with the tails. The heads, compared to the tails, only take up a little amount of space.
Yeah.
So they can easily get past the heads, and then they are quite nicely move through the tails, and then you can move straight through.
Okay.
Whereas the other things, they like the heads, but then they can't get through the denser part of the tails.
Okay.
Does that make sense?
Yeah. So with phospholipids wouldn't dissolve in water then?
No. So they don't classify as hydrophobic or hydrophilic. I mean, you could say it has a hydrophilic head and a hydrophobic tail. But what it's usually termed as is amphipathic. Amphi coming from ambidextrous. It's kind of spelled the same. So ambidextrous is both hands, so amphipathic. It fears both things.
Okay.
But yeah. Or in other terms, loves both things.
Okay. So then what would be the similarities between the two?
So the similarities would be those fatty acid tails that you just talked about. They're considered fats because they're under that section of the three main types of fat. So they're fats. They have fatty tails. Give me a couple seconds. I'm trying to think if phospholipids do have a glycerol in them as well.
Phospholipids, they do.
There's a structure right there. Do you kind of get the similarities and the differences though?
Yeah, I think so.
So basically the similarities are their category and that they both have hydrophobic tails. Then the differences are the glycerol versus the
nitrogen.
Yeah, phosphate, sometimes nitrogen and oxygen, and the 2 to 3 tails.
Okay. Where do we find most of the phospholipids in our body?
They're going to be in. So you saw that thing that I was just drawing right there? Imagine
Yeah.
I just didn't erase
Just-
it.
just
so it's
so-
the cell walls.
Yeah. Okay.
So that's where you're going to find the phospholipids.